I will start with the general introduction to phylogenetic function inference. So what is this about? And then introduce you to the basic features in PhyloGenes, followed by a case study of using PhyloGenes for inferring gene functions. At the end, we will cover how to find the data and navigate to the PhyloGenes website using a live demo. So from sequence to function, the fastest way is to run a computational prediction, which can then guide you to design the proper experiments to validate the function. The most commonly used methods are sequence similarity based, so everyone uses BLAST and also interpro-scan. Another type of methods is to consider evolutionary history of function, using the evolutionary history in function prediction. This is referred to as phylogenetic function inference. Going to show you a toy example. Here, the evolutionary history of a group of homologous sequences from human and mouse are represented in the form of a gene tree. And any pair of genes are related to each other either through speciation or duplication. So here, the green dots indicates a speciation event. The yellow dots, duplication event. Orthologs are a pair of genes or a pair of homologs that are related by speciation. Paralogs are related by duplication. Paralogs can be within a species, such as this one, or between species, such as this one. The underlying principle that supports phylogenetic function inference is the belief that, showing here, protein functions changes rapidly after duplication, leading to paralogs with different functions, orthologs with their original ancestral function. So in another word, orthologs share the same function. So using the same example, let's say that we know the functions of the mouse gene 1. We also know the function of mouse gene 3. And then applying the ortholog conjecture, we can be more confidently to predict the function for human gene 1, because that's an ortholog of the mouse gene 1. And we can predict the function for human gene 3. As for human gene 2, we need to be more careful when we want to infer the function. We probably have to look into additional information because right now it's a paralog from the mouse genes. Okay, so a quick recap. Phylogenetic function inference is to assign function from a known gene to an unknown using evolutionary history of the genes. So there are two key points here. We need to identify the orthologs, and we need to know the function for at least one of the orthologs so that we can use that to infer the function for the others. Okay, so so far, any questions?
There are no questions in the chat besides trying to figure out the sound. Thanks, Pei-Fen.
Okay, so but if you don't want to type, if you have a question, you can unmute for the time that you wanted to speak up for the questions if you have. Okay. So if no, let's continue. So PhyloGenes is a free online resource for phylogenetic function inference. So you can see the URL on the top. It displays phylogenetic trees of gene families. So these are pre-computed gene families from many species. Alongside a gene tree, it also displays experimental gene function information. The goal is to assist plant scientists to infer functions for the unknown. So currently, there are 40 plant genomes included in the gene families. In addition, we also included 10 non-plants. So these are well-studied model organisms that have most experimentally characterized gene functions. So this is a typical gene family page. It has 3 parts. The gene information band shows the meta information of the family, such as number of genes, list of species that are included in the family. You can follow that link. And the taxonomic range of those species. So in this example, it says LUCA. That's for last universal common ancestor. So basically, it says this family includes all living organisms. The left panel is the tree, is the gene tree. The right panel shows gene information, such as known function, sequence, gene ID, etc. Each gene row in the right panel is aligned with the corresponding gene in the gene tree. The gene trees in PhyloGenes, they are reduced form of gene trees in Panzer. Panzer families are constructed by the unique Panzer pipeline. So there are 142 genomes currently included in its release. As I mentioned, the PhyloGenes gene trees are reduced version of the Panzer gene trees. So genes from non-plants, other than the 10 model organisms, they were removed from each Panzer family, producing a plant-centric view of the family in PhyloGenes. So if you used Panzer before, you are familiar with their data, and when you come to PhyloGenes, you are seeing a reduced version, which is plant-centric. So here, just to highlight some basic features on the gene tree if you come to PhyloGenes. We distinguish speciation and duplication. So again, green dots are speciation nodes, yellow dots or orange dots are duplication nodes. And we also distinguish subfamilies. So after a duplication event, the faster evolving branch here, you can see after duplication, the faster evolving branch indicated with a longer branch length, it became a new subfamily. Each leaf node on the tree is a gene. It's labeled with the gene name and the species name. If there is a common name for the gene, we would show that. Otherwise, we display the gene ID. You can collapse any internal nodes. A collapsed node is labeled with the taxonomic range of all its children genes. Okay. And then in the gene information table next to the tree, we display known functions. So these are Go molecular function annotations supported by experimental evidence. So this, all the annotations were curated by many communities, including CHAIR and other communities. They are aggregated in the central GOA project, and we retrieve the data from GOA. Each column is a Go term that is annotated to at least one gene of this family. A yellow circle here indicates the presence of that function for that gene. You can click on the yellow circle, which will let you access to the details about the annotation, such as Go term definition, evidence description, and publications that supports the function for that gene. So I'm gonna to pause. Any questions so far? If I speak too fast, let me know. We can slow down. Okay. So if no questions, we'll continue. I'm going to use an example case study to show how you can use PhyloGenes to infer functions and explore the evolutionary histories of the functions. So here, we pick a grape gene. We look up its function in UniProt, which says catalytic activity. So basically, it tells you it's not enzyme, but no further information on what kind of enzyme. We can look up the same grape gene in another resource. Here, we found specific enzyme activities annotated to this grape gene. So here, fatty acid-CoA ligase and for coumarate-CoA ligase. So these are two different functions. Then does the grape gene have both functions or just one? Which one? How do the two functions relate to each other? We can find that kind of information from here. So then we look up PhyloGenes. We found the gene family that has the grape gene shown here. As expected, there's no characterized function for this grape gene. However, its Arabidopsis ortholog is a fatty acid-CoA ligase, right? Using the ortholog conjecture, we can then infer the function for our grape gene. It's more likely to be a fatty acid-CoA ligase. We also found in the same gene family that another Arabidopsis gene has been characterized as a 4-coumarate-CoA ligase. So similarly, we can infer the function for the second grape gene in the family as more likely to be a 4-coumarate-CoA ligase. Okay. So the beauty of the gene tree does not stop here. Relying on other genes that are characterized as fatty acid-CoA ligase in the same family, we can trace back to the most recent common ancestor of that enzyme activity. So similarly, we can also trace back to the most recent common ancestor for coumarate-CoA ligase. And by looking at the gene tree, it indicates that coumarate-CoA ligase evolved from a fatty acid-CoA ligase after this gene duplication event. So that's clear, everyone able to follow? Okay. So now I'm going to switch for a live demo. Okay. Let's go to, so you come to the homepage of PhyloGenes, and you found that there are two search boxes. You can use either one. You can search the PhyloGenes website by gene or by a general keyword. Just wanted to say that currently no partial stream match or wildcard is supported for search, so it's only returning exact match. But all search returns gene family. Usually, if you search for a gene, it will find the family that has your gene, then will take you directly to the gene family page. If you search by a keyword, you are more likely to get a list of matching families. So in our demo, let's say we're interested in cytochrome P450, so you could type P450. And then there are 29 gene families found. You could filter the results by using either the taxonomic range of the families or species included in families. Let's say that we're interested in families that are specific to land plants. So we could check Umbreophyta. Then we get a shorter list of families. Then you could go to any one of it. Let's say we're picking the second one. You could hide the legend. So by default, we're showing a tree in its compact view, which means that only those functionally characterized genes are displayed, indicated by a yellow flask. All other genes are hidden. But you could expand all the hidden nodes, so all the collapsed nodes, by click on expand all. So now you notice that this family has over 500 genes. The tree is large. It's very hard for us to find what we're looking for. What you can do is that you go to this operations menu. You could do prune tree by organism. Let's say that I'm only interested in Arabidopsis and Chinese cabbage. I would deselect everything, only select Arabidopsis and Chinese cabbage. Then I can update the tree to get an even slim view of this gene family. Okay, so now we see a slim view, so you could even expand here. So it led us to focus on just a subset of genes, right? The genes from Arabidopsis and the Chinese cabbage for this gene family. So we heard people complain that they can't find their genes. Sometimes it's probably a typo, or sometimes maybe you only put a partial string, but it could be legitimate. Keep in mind that not all genes from a genome are part of gene families. Alternatively, it could be that the species of your study is not yet included in PhyloGenes. So for that, we go back to the homepage of PhyloGenes. You could check the species list here. If you don't see your species of study, you could follow this button, which will take you to a tool called Tree Grafter. So Tree Grafter, you could copy paste your protein sequence. It will run a program, will try to find the best matching gene family, and then place your sequence to the proper position in that gene family tree. So let's say that I am interested in, I have a sequence which is from the mint plant. Then I would copy paste that sequence into this window. And then I run grafting. Tree grafter is very computationally expensive. Usually it will take a few minutes to run. So yeah, be patient and only use it that when you are sure that your species of study is not in PhyloGenes. So hopefully it will return our results soon. Okay, so again, we can hide the legend. So you can see that my sequence is labeled on the tree as grafted. So I wanted to point out is that if you go back to the tree grafter page, let's see. There's some description here. After this demo, you could maybe read the published paper about the algorithm. But what I wanted to say is that let me just bring up a saved image of the tree grafter results so that we don't wait for another couple minutes to see the results. So this is an image. What I wanted to say is that tree grafter was developed primarily for function inference, which means that it lets you to propagate function to the grafted sequence from its ancestor if the ancestor has a known function. The tool was not developed for you to tracing the evolutionary history of the grafted sequence. In this example, you can see that I grafted a mint sequence, right? It's been placed under this duplication node of monkey flower. It's very unlikely event, very unlikely evolutionary event. So when you use tree grafter, keep that in mind that the purpose is for function inference, is not for evolutionary history inference. Okay, and since we're here, I want to say that one apology. We are experiencing a software glitch for this tree grafter tool. So it only works if you are using Chrome as your browser, and if you are using so you go to here, you are using a private window. So you go to Chrome and then you use private window. That will work when you run the tree grafter. At the moment, we have this limitation. We're working to resolve it, but if you really wanted to try out it, make sure that you use Chrome and then you open a private window. Okay. So that concludes my presentation. For the remaining time, I can answer any questions if you have. When you speak up, be sure to unmute yourself or type your question into the chat window. Actually, before we go to the question and answer section, I wanted to bring up this URL. So this is, if you could please save this URL. It will take you to a very short survey. And we will be very grateful if you can fill out the survey it helps us to prioritize features to be included in our future releases. So that URL, please save it. And if you wanted to see today's presentation, you can also go to this URL to get a copy of today's presentation, my slides. Okay, so thank you for listening. I'll be happy to take any questions.
Peyton, we have two questions. One from Marina, who asks, in the case of genes with multiple names, are all the names in the platform, or does each gene only have one name?
That's a very good question. So let's go back to okay. So that information is here. So we go to either you go to about or help, it will take you to the same place. So we go to, it's our help document, and you can go to user guide, go to search. It will tell you, what is allowed for search. But in short, gene name, if we're talking about common name, it's what usually there's only one common name stored in our database. Well, you can read here that tells you that all the names and IDs, they are really coming from UniProt for each of the protein entry. So that's probably very brief answer to your question. So usually there's only one common name included that you can use to search, or you could search by gene ID. But if you still don't find anything, I would suggest to you, it's always the best to use UniProt ID. So if you can, find the UniProt ID for your gene, use that. Usually you have a better chance to find your gene.
Okay, the other question from Chuck was about being able to use wildcards in the search window, but I think your answer your answer to the previous question in this page addresses that.
Yeah, so I mentioned briefly, probably it's not clear that wildcard is not supported, partial string is not supported. So for example, that your gene symbol is FLC, right? If you only type FL, it will not find it. So that's a feature that on our to-do list, we will work on it. But in terms of priority among all the features that we wanted to do, that's why I put up this survey form. So we wanted to work on the features that will be helpful to more users. So please fill out the survey form that will help us to prioritize what feature to release in the future.
We have a question from Ajar saying, if I have a sequence from banana that is not included in PhyloGenes, how can I find a similar gene?
It's only what you spend a third of the time on.
So I would think banana is one of the genome included in PhyloGenes, I believe, that we could see. Well, we could see the species tree. It's a monocot, right?
It's there. It's
Yeah, it's here, right? So it's a genome included in the Panther pipeline to build the families. But if you don't find your gene, I would think you could still try using the tree grafter, because tree grafter, it does not ask which species your sequence is from, right? You could still try to run it. It may or may not find a best matching family. It really depends on the algorithm. But you could still try using the tree grafter too.
And that's it from the questions in the chat window, Peyton.
Okay, great. So yeah, Thanks again for coming to the webinar. I hope that that's helpful today. But if not, you could, always, click on the help or contact us, email us, send us your question. And please, if you could fill out the survey form, that will really help us to understand your needs and will help us to prioritize future releases. Thank you very much.